3 folder. We also have papers talking about genetically modifying mammalian cells and even human beings and monkeys. So we have stuff all about all different types of organisms. Make sure you read those papers. Now, normally when you're looking at papers on genetic modification, especially animals and humans, what you're more concerned about is the methods, because that's usually the major part that you can apply to your own research. So read the methods carefully, understand the methods of how this stuff is done, and that will be really applicable to your research and the stuff you want to do in your future research. Remember, next week, or not next week, but in 2 weeks, George Church is going to be giving a class. He's going to talk about genetic engineering for a short bit, and then he's just going to answer your questions. So make sure you bring some questions for George Church in 2 weeks, and you're ready to ask him some good stuff. And he's ready. I talked to him. He's ready to answer whatever you can throw at him to make sure you got something good. So to start off this month, we're just going to go over some basics on genetic engineering, and then we're going to do a little work with the computer software SnapGene. So what you should all do today is download SnapGene. We're going to do a brief tutorial on that. So I'm posting a link. If you can't see the link, what you need to do is change top chat to live chat in the top left-hand corner of the chat, of the YouTube chat. Change it from top to live, and you should see the link for the SnapGene viewer. Download and install that, and we'll go over that a little bit. So while I'm rambling, make sure you do that. All right. So genetic engineering, the most basic point of genetic engineering is you're just trying to put DNA into cells. That's it. Just put DNA into cells. You can take DNA from basically anything. So you want to take DNA from a rabbit and put it in a human. You want to take DNA from a human and put it in a jellyfish. You can do whatever you want. Now, it doesn't necessarily mean it's going to work, right? Because the genes, the metabolism of these organisms are completely different. The genes they interact with are completely different. But if you wanted to, you could put the DNA from any organism into a cell. Now, generally, the way you get this DNA is you order it from a company. So there are a lot of companies who do DNA synthesis. The main ones we work with and that I recommend are GeneWiz. So genewiz.com is one of them. Here's a link. And there are a number of other companies you can find. GenScript, Elim, IDT, if they'll let you order from them. And from these companies, what you can do is you can have DNA synthesized or made. So if you remember back in previous lectures and things, maybe it was last week or 2 weeks ago, last lecture, where we looked at genes using the NCBI database. You can find genes through the NCBI database that you want to use and just have these companies synthesize them. So they'll just make them synthetically. The cost of these genes is generally around 10 cents a base pair. So 1,000 base pairs will be like $100 or $150. 3,000 base pairs will be like $300, etc. But generally, you don't use genes by themselves, right? So genes need to have a sort of context. and this context is other DNA elements that help these genes being made. Now, when those genes are put into a context and used for genetic engineering, we usually call that a plasmid. Now, a plasmid is a circular piece of DNA. It doesn't like look circular physically generally. It's just circular in construction. A linear piece of DNA would have ends broken, right? It would just be like linear ends broken, and a circular piece has ends connected, right? And the ends connected make it so that it can be replicated easy. Cells can take it up and use it. That we call them plasmids, right? These circular pieces of DNA that have context that organisms can use. What's the basic process to synthesize? To synthesize DNA, uh, from a high level, basically all they do is they just use chemical processes to connect nucleotides together one at a time. So they'll just be like A, add some chemicals, T, add some chemicals, G, add some chemicals, etc. It's more complicated than that, obviously, but not too much more complicated. It's basically machines that do that. Yeah. So when you're doing genetic engineering, a plasmid is basically the building block of genetic engineering. It's what you're always going to be using. It's what you're always going to be talking about. It's what you're always going to be working with. Is what you're always going to be ordering. In the case of month 3, we gave you plasmids to put in bacteria that make the plasmid makes the bacteria fluoresce green. So it has this protein GFP. GFP, the green fluorescent protein, is from a jellyfish. Now we have the same thing for a yeast, but the thing is you can't take the bacterial plasmid and put it in the yeast. It won't work because there's different elements that are required for different organisms, right? So bacterial DNA won't just work straight up in yeast or other organisms, and yeast DNA won't work straight up in other organisms. Now, generally, when you start to get to higher mammals, DNA can be pretty translatable between the different species. So if you do experiments on monkeys and humans, you're probably going to use the same DNA. Even if you're doing experiments on, say, like horses or dogs, there's a good chance you're using similar or same DNA. We all have similar genetic systems and metabolisms that are pretty compatible, so we can do that. Now, these plasmids, don't mix up your chocolate plasmid with peanut butter. Exactly, Craig. Exactly. I have no idea what that means. But so basically, your goal with any sort of genetic engineering is just to get DNA inside of a cell. That's your number one main goal. Get DNA inside of a cell, right? And once the DNA is inside of the cell, then the cell will be convinced that it's its own DNA and it will start to try to use it. Getting the DNA inside the cell is usually the hard part, the most difficult part. Alpha Centauri says, so basically DNA or plasmids are mostly compatible within their major branch of life. Kind of. I mean, there's always exceptions. There's always different things that are going on. But generally that's probably true. You could probably use like a yeast plasmid and fungus or something like that, and a bacterial plasmid across some bacterial species. But it gets complicated. But yeah, that's a good general motto. But so where was I? What were we talking about? Oh yeah, so he sent you these plasmid DNAs that express these green fluorescent proteins. What we also sent you bacteria that already have plasmid in it, these P dusk, P dawn systems that are really cool. They're these light controlled biosensors, and there's this month that you can read about them. And we're going to look at those plasmids today using SnapGene. All right. So if you haven't downloaded SnapGene already, um, make sure you do right now, and we'll demonstrate it and get into it a little bit. There are no limitations to the size. So there's the question, is there any limitation to the size, number of base pairs of DNA you could put in plasma? No. So theoretically, there's not. The problem is you have to start thinking about physically, right? So if you're trying to put together a circle that's really big, right? So say you have a rope and you're trying to connect two ends of a rope. The larger the rope, the more difficult it is to connect those two ends of the rope, right? Because you have to find them and unwind them and put them next to each other. So as you get bigger and bigger pieces of DNA, it becomes more and more complicated to work with them, to engineer them yourselves, and things like that. So generally, plasmids are around 10,000 base pairs or less, but you can easily get plasmids that are 15 or 20,000 base pairs. Bigger plasmids, generally called bacterial artificial chromosomes, or BACs, BAC, are also used, and those can be really big. Uh, but generally, those aren't for genetic engineering. Generally, those are used for DNA storage. If you're doing, like, they used to use them for DNA sequencing a lot, and other things to move large genes around between different organisms. Um, is there no way to add primers throughout the line of code? Me, myself, I'm not quite sure what that question means. So yeah, I don't know how to answer it, but yeah, if you word it differently or ask me again later, or ask me offline, could you design a plasmid? Yes, you can design plasmid. You can order the DNA, you can have it synthetically. So Esther says, how difficult is it to transform a large plasmid versus a small one? Generally, it doesn't matter too much, because when you're doing genetic engineering, you don't care so much about efficiency, right? So if I'm trying to engineer a bacteria, if I have a big plasmid, the efficiency is going to be a lot lower, but it doesn't matter if it works as long as I get some bacteria that's transformed. Now, if you're talking about like human genetic engineering or like a gene therapy, it matters a lot more, because what happens is you're trying to get the most amount into cells possible, um, into your cells. So a bigger plasmid is going to be a lot more difficult to do that. So generally, a smaller one is better, but how much is unclear. If the plasmid is really big, over 15 or 20 kilobases, it matters a lot. If it's under 15 or 10 kilobases, the papers and data out there are pretty unclear how big the effect would be. Come and See says, can you talk about the problems with using bacterial plasmids with regards to adverse immune responses triggered by bacterial antibodies and fragments left over after purification? Ugh. I don't know if that's really in the scope, but so generally, our immune system, there is DNA is different between eukaryotes and prokaryotes, right? There's just differences. There's differences in methylation and differences in other things that happen to the DNA between eukaryotes and prokaryotes. Now, eukaryotes are like mammals and bigger organisms, and prokaryotes are like bacteria, right? So they're more primitive organisms. Now, generally, when you're replicating plasmids, you replicate the DNA in bacteria, because bacteria like to replicate DNA, so they're an easy way to make large quantities of DNA to use for gene therapy. Now, generally, if you put bacterial plasmid DNA in your body, you're most likely not going to have an immune response, even despite the methylation differences. There can be. Some people say there are, and there's gene silencing and other things, but the data is really it's not like there's probably differences people show are like twofold or something like that. Twofold is not a big effect in biology. It's very, very, very small, um, because that can easily be overcome in many different ways. Adverse immune response triggered by bacterial antibodies for fragments left over after purification. Now, generally, gene therapies are purified extremely well, so there shouldn't be any fragments left over. If you were, for instance, to say inject yourself with improperly purified DNA, and you had some of these, what they call LPS generally, or endotoxins, left over from the purification process in bacteria, um, what would probably happen is you'd have a slight immune response, if you're not immune compromised. Immune compromised people probably have something different happen, but there are easy to and inexpensive kits you can purchase where you can test endotoxin levels using horseshoe crab blood. Yes, horseshoe crab blood. It's really interesting. Their their blood coagulates, um, based on the amount of endotoxin in it. Uh, who would have known? So yeah, they purify horseshoe crab blood and use it to test the amount of. Can you clone that blood? I don't know what that means. The crabs clone it, so they're pretty good at it. Um, so that's how people generally get it. But yeah, so that's that. So we're gonna do SnapGene. So if you haven't downloaded SnapGene, download it and install it right now. Download and install SnapGene. I'm going to share my screen or share my program. All right. SnapGene. SnapGene visible? Yes. All right, great. Seems to be visible. Yeah, SnapGene Viewer. SnapGene Viewer. Just use SnapGene Viewer. You don't need um, the other version of SnapGene generally, and it's pretty expensive. So just use the SnapGene Viewer. It's totally okay. Now, what we're going to do today is we're going to look at one of the plasmids we're using in month 3. So we're going to go to import. We're going to go import add gene plasmids. Now, add gene is a plasmid database that's only available to academics generally. But if you work with a biohacker space, you can generally get DNA. You can generally get the biohacker space to request DNA from AddGene, and they will provide it. Um, I went down? What do you mean? There's nothing on the screen? Okay, that's fine. I just opened up a dial. Like when you click on import. So what you want to do is when you click on import, I know it's because I AddGene. So when you click on import, type in the word pdusk, PDUSK, and it will import pdusk for you. All right. Now let me know if the screen pops back up. All right, you got it. All right. Still down. Still black. Maybe I have to screen share my new screen. You're live on. All right, let's try this again. Can you see it? Oh, there it is. All right. Can you see it, everybody? Yes? All right. So pdusk. Type it in, get it, and download it. What you should see is what you see on the screen. This is called a plasmid map. Basically, it provides a bunch of information about the plasmid that helps you understand it and genetically modify it. Right? So we're just going to take a minute and let you all catch up. Got it. Yes, good job. Go to add gene. So when you go to import from add gene. PCR should work on any DNA. So yes, it works on plasmids. Plasmids are DNA that's just circular, right? So, and what you're staring at on the screen, this plasmid map, what we see is just a circular piece of DNA. It's just a representation. Um, all right. So let's go through this. All right. So this just gives us a large representation of the plasmid. What we see, all these little weird letters and numbers, these are restriction enzymes. Now, don't really worry about this stuff right now. In the Biohacker 102 class, we're going to go over this a bit more and teach you how to use restriction enzymes to engineer your own DNA. But suffice it to say is that restriction enzymes are specific enzymes that people have found that will cut DNA at specific places. What you can do then is you could cut DNA at specific places and glue it back together. And by doing that, you can put together basically whatever DNA you want, right? You can't find add gene. So when you open up Snapgene, the initial thing, you should click on import, and there should be like a drag down on the import button, and you should go to add gene plasmids. Thank you, Matt. Appreciate it. So this is one of the plasmids we're going to be working with, the P dusk plasmid. Now, it's a bit complicated to understand how it works if you don't know what's going on, but we'll try to give a brief overview. Now, what the arrows indicate is they indicate the direction that the DNA is being translated. So, what this means is that generally, when a gene is being made, when there's a protein being made from a gene, it has a direction, right? So, you have a top and a bottom strand of DNA. Now, the DNA can either be read from the top strand or the bottom strand. Now, depending which strand it's read from, that determines its directionality, so to speak. There's right? So this directionality then is how what leads to how the gene is being read and turned into a protein. So that's what these arrows directions are. Um, it's not important for our context right now, so don't worry about it too much. But that's just to help you understand this a little bit better. So now what we're going to want to do is we're going to want to click on sequence at the bottom underneath the plasmid. And what this does is this gives us a view of the actual DNA sequence of the whole entire plasmid, right? So because it is circular, you have to understand that the end connects to the beginning. It's not linear, it's circular. So the end connects to the beginning. So if you wanted to say, uh, create some DNA that connected with this or was a PCR primer or something, the sequence at the beginning and the end are together, right? Now, there are a few important parts of the plasmid that you should always look at and always understand. The first is antibiotic resistance marker. That's usually one of the biggest parts of the plasmid. Now, antibiotic resistance marker, we through the last class we used antibiotics or the last month to grow our PJE202 uh, bioluminescent bacteria. Now, what antibiotics do is they kill all the bacteria that don't have the plasmid DNA, right? But allow the bacteria that have the plasmid DNA to survive. Now, this is key because bacteria just multiply and grow so fast. What you need to do is you need to remove all the ones that don't have this. Now, understand this is commonly used in molecular biology, super commonly used. But also understand it is contributing zero to antibiotic resistance in the world. The amount of antibiotics that are used through these molecular biology techniques sorry sorry about that. Oh, I need a drink. The amount of antibiotics that are used are minuscule compared to the amount that are used to feed agricultural animals and also for human beings. It's very minuscule. We're working with milligrams per liter generally, right? Milligrams is really tiny. So when you what does he say? Puff, no pass, bro. I know that's my fault. I'm sorry. All right. So what you do is if you get a plasmid and somebody sends you a plasmid and the plasmid map, the first thing you're going to want to know is what's the antibiotic resistance, right? And so you look for these giant antibiotic resistant markers. This one says CAN-R. It confers resistance to kanamycin in bacteria or G418 in eukaryotes. So we're using bacteria, so it confers resistance to kanamycin, right? So when we do experiments with P. dusk and P. dawn, we're going to use the kanamycin antibiotic to make sure we select only for bacteria that have been genetically modified with this plasmid, right? Now, the next important part of the plasmid, which you don't need to know much about, but you need to know exists, is what we call the origin of replication. Now, origin of replication just basically tells the cell that it's in that the cell should replicate this piece of DNA. So, like if the cell, uh, if a new cell is created from the cell, right? So, if the cells are growing and multiplying, they replicate the plasmid and share it between their daughter cells. So the plasmid keeps growing and keeps being replicated. So this origin of replication is common in bacteria and yeast generally. Not so common in other cell types, but very common in bacteria or yeast. Now, this origin of replication can determine how many copies of this DNA are also in the cell. So there are high copy number plasmids and low copy number plasmids. Now, say if you're making a gene that can be harmful to the bacteria or yeast, you want to use an origin of replication that is more low copy number. So it doesn't make a lot of the gene. Whereas if you're making a gene that you just want to make the most of, you're almost always going to use a high copy number origin of replication. And you can find out more information about origin of replications online, but by just putting your mouse over the origin of replication in SnapGene, it tells you this origin of replication is a high copy number, PUC origin, PBR322 origin, PMB1 origin, right? So generally, some of these origins have similar ancestries and similar and different names. Now, the final important part of the plasmid is what we call the multiple cloning site. MCS, multiple cloning site. Now, generally, the multiple cloning site is where all the restriction enzymes are. So when people create plasmids, or if you create a plasmid from scratch or something like that, generally what you do is you want to give people the most amount of options to engineer this plasmid for their own use. So you put in a bunch of these different restriction enzyme sites, right? And what you can see is that there are a bunch, and the majority of these restriction enzyme sites, they isolate around this region right here, right? There's some everywhere because you're going to have, it's impossible to avoid all restriction enzyme sites, but these ones kind of congregate right here. And we call that the multiple cloning site, the MCS, or multiple cloning site. When you're engineering DNA in plasmids, you generally call it cloning, and it's different from the cloning that people think with like human cloning or animal cloning. It This cloning refers to just creating new copies of the DNA, right? But we call it cloning also. Scientists call it cloning. I didn't name it. Don't blame me. I know it's confusing, but it's called cloning. So if you hear somebody referring to like DNA cloning or plasmid cloning, it's generally referring to plasmid genetic engineering, like engineering the plasmid to do what you want. So you get a cell immune to a particular antibiotic, make transgenic, and then use that antibiotic. Yeah, kind of, Christopher. So basically, what this allows is it allows you to select antibiotics, or selection, or different selection things allow you to select. Um, so if say I'm trying to engineer a bacteria and I do a transformation like we're going to do in month 3, and you put the DNA in the bacterial cell, you have to, not all the bacteria are going to get DNA inside them. So you have to have a way to isolate the bacteria with DNA inside them from the bacteria that don't have DNA inside them. So what you have to do basically is select out all the bacteria who don't have DNA and keep all the ones that do. And antibiotics are a good way, or these selection things. Generally, I think they still call them antibiotics, even when they're used in yeast and mammalian cells and all these other things, even though antibiotics generally refers to bacteria. My screen is black. There's nothing there. All right, well, just come back to me. Stop sharing. I'll answer some questions. All right. Am I back? Not yet? All right, I'm back. Do the placement, come and see says, do the placement of these important sequences fall in some pattern? They kind of do, but not necessarily. So they've found that like having the origin of replication on the opposite side of plasmid from your gene can lead to more copies of the plasmid and things like that, higher levels of gene expression. But generally, plasmids are just a mess. There's probably so much DNA in all plasmids that is just junk. I've analyzed them myself. I've created plasmid from scratch myself. A lot of it is junk in there. But generally, it's difficult to tell what is junk and what isn't junk. So I generally would not mess with that if I were you. But understand that all the DNA in a plasmid are not necessarily there for a reason. Just remember that. Keep that in mind. Take that how you will, but just understand that. Like, you have to understand that a lot of plasmids were created a long time ago when DNA genetic engineering first started, and people just basically sloppily put together what they could because they didn't have all the restriction enzymes and all the genetic engineering techniques we do now. and we just kind of stuck with it because scientists are lazy. They are really lazy. Um, Dave Hansen asks, don't all cells have DNA in them? Yes, they do. All cells do have DNA in them. But what a plasmid does is the cell thinks it's its own DNA because of the different DNA elements in there, right? So, like the origin of replication, when you put a piece of DNA with an origin of replication inside a bacterial cell, the bacterial cell immediately thinks it's its own chromosome, right? Bacteria can't think. They can't look at a piece of DNA and say, oh, that's not my chromosome. No, they look at the piece of DNA and says, does it have an origin of replication? If the answer is yes, it thinks it's its own DNA, right? It's pretty simple. They're pretty simple organisms, so they're easy to hack like that. Can you replicate plasmids on a large scale? Me, myself. Yes, you can. You just grow up a lot of bacteria and you purify it from the bacteria. That's how you replicate it. Scientists generally always use bacteria to replicate DNA because they grow the fastest, they grow the largest, and they make DNA really, really well. Joel asks, how do we identify the MCS in this map? When you're trying to identify the MCS, you're just looking for, number one, where a lot of restriction sites are. And then number two, what you can look for are certain promoter and terminator sequences. So a promoter is a region that encourages the DNA in front of it to be made, and a terminator tells uh, where the DNA where it should stop being made. So, for instance, if you see a piece of DNA labeled promoter, you see a piece of DNA labeled RBS or terminator, you're generally in the region where the mm, um, multiple cloning site is going to be. But you have to understand multiple cloning site is not necessary. You could technically clone DNA into the plasmid wherever you want to. Um, any restriction sites. As long as you put in the correct DNA elements, you could put DNA anywhere in the plasmid you want. As long as you don't like cut the origin of replication or the antibiotic resistance or something like that, you're pretty good. You can do whatever you want. The multiple cloning site is just made to help you, to give you a lot of options. But nowadays it matters not so much. The RBS stands for ribosome binding site. So if you remember from month one, we talked a little bit about ribosomes. Ribosomes are what make proteins, right? So a ribosome binding site is the beginning of a piece of DNA where a protein is going to be made. So it's more of a beginning region than an end region. A terminator is an end region, just like it sounds. All right, so yeah, I would suggest mess around with SnapGene. You know, uh, go to pull up the add gene import and just import some random plasmids, or go to the add gene website and search for plasmids you might be interested in and import those and look at what they're composed of. Look at the difference between some human plasmids and some yeast plasmids and some bacterial plasmids. Uh, we'll even send you, yeah, well, one thing I think we forgot to do is I can do it right now. I'll put the DNA sequence for the yeast and the bacterial plasmids in our month 3. So you can look at the DNA sequence for the yeast and bacterial plasmids. And you can analyze those. Those are the plasmids you're going to be using for the experiments we do. So there's that one. Let me see. Got any other questions? All right, so I put the plasmids in the folder, month 3 folder. So you should be able to find them there. So you can look over those. Humans have plasmids. You can use plasmids on humans, but humans don't have plasmids naturally, not human plasmids. If you got your genome sequence, could you open up the raw data? You could open up the raw data in something like SnapGene, but your genome is way too big. You're talking terabytes of data. So you wouldn't be able to open it up per se and peruse the whole thing because it's just so massive. Any other questions? If not, you all know what time it is. It's brief time. From now on, we're going to do classes in one shipment. So we decided that we're going to shorten the classes to 2 months and have classes every week instead of doing the every other week thing. I think it'll be a lot better to do every week. And we're just going to send out one package then with everything in it from now on. Because obviously you guys can tell it's been a total mess trying to send you guys everything all the time. So yeah, so 102, the supplies will be sent out in one shipment. And yeah, we're going to do it again. We're going to do the classes every 2 months, starting in May. So starting in May, we're going to do the class every 2 months, and we hope to add some more classes in the future. Classes on working with plants, working with animals, even maybe doing some longevity experiments and tests. So Alpha Centauri, no, you can make your own plasmid from scratch. You have to get the pieces of DNA synthesized, and then you just stick them together. So yeah, I've created my own plasmid from scratch. It's totally totally cool. It's a little bit more complicated. Can you sign up for 102? Yeah, you can sign up for 102. It's on the website. Feel free to sign up. It's going to include a boot camp program on site. Nothing on site right now. Sorry. Hopefully, we're moving into a bigger space. Maybe someday, but one day. All right. I think I'm gonna pass it over to Esther, and she's gonna give you some demonstration explanations today. So get ready for the brief. It's on you, Esther.
Burp. Burp, burp. Oh man, am I on?
Yeah.
Can you hear me?
Oh, you're gone. Now you're back.
Oh, I'm back. I'm on. I'm back. I'm on.
Just click on yourself.
Yeah. Alpha Centauri, you saw two prices. You saw $200 and $600 for the 102 class. So if you took this class, that means it's actually $400. My bad. There's
3 prices. 200 if you already have everything.
200 if you have everything. 400 if you have some things and you're like not part of this class.
No, if you are part of this. 400 if you took 101.
All right, 400 if you took 101.
600 if you didn't.
600 if you didn't take shit. So, yeah, those are the different prices. And yeah, I would highly encourage you to sign up for another class because we go really in depth on how to design your own plasmids, which would be super awesome. Yeah. Can you guys hear me? Do you still hear an echo? Echo, echo, echo?
Turn off your own volume.
Okay. I think we're good. All right. So we're going to be doing a lot of calculations today. Maths. I know. Everyone fucking hates maths. So we're going to use a calculator because I hate doing math. Part of being a scientist. So take out your calculators. And so we're at month 2. We finished, or you guys are going through bioluminescent bacteria kit. And I saw some great streaks of how to streak bacteria. Some prime shit out there. I'm really proud of some of you, most of you, almost all of you. I know, Craig, we have to use maths, but there's no way around it. Unfortunately, when you're a biohacker, you have to source every little thing from agar to like LB broth and shit like that. So I want to teach you something valuable, maybe, so you don't have to order pre-measured tubes from us. I mean, you can totally get pre-measured tubes from us, but it'll be kind of costly. So I'm going to teach you how to do your own calculations and to make your own media at home. All right. I'm going to screen share. Screen share. Share. How to make a media.
Screen share something.
I know. Okay. How to make a media.
All right. See you guys tomorrow.
Google Hangouts. Hide. Oh my God. How to make a media. We're good? We're good? Okay. You guys are going to have to tell me.
Are they going to present your life?
I'm going to present. I'll probably take questions at the end, but I'll go really, really slowly. I can't really see your questions right now because, yeah. So how to make a media. There are different types of media. There's all these media, and got it from Wikipedia. There's culture, minimal, selective, differential, transport medias. And so what you're generally working with is culture media. You are culturing bacteria, you're culturing yeast, you're growing up things inside of media. Minimal media is pretty much the bare minimum things that you need to grow on, such as like glucose, which most organisms need, and probably like very minimal, like, what is it called? Nutrients, things like that. Selective media is things that you want to be selecting for. For example, we talked a lot about antibiotics, and adding antibiotics to the media makes it selective, meaning I want the P. dusk to grow with the kanamycin resistance. So I'll probably put kanamycin in one plate and then leave out the kanamycin in the other plate, which makes it selective. So that's what a selective media is. Differential media is interesting media that is kind of used for very specific, like highly specific things. For example, like blood agar media for like cultivating human cells and things like that. So that's kind of a different beast on its own. And then there's transport media, which is media that you use to transport different types of organisms. So liquid versus agar. So we sent you in month 2 the bioluminescent bacteria kit. This included LB ampicillin broth, and also LB ampicillin agar. So the difference is the broth is just the liquid culture, and the agar is on plates. So it's pretty easy to differentiate. Okay, are you guys ready for calculations? Yeah? Blood media. All right. Calculations. Okay, so we need to make 1% weight per volume LB agar. Generally, it's 1%. This is used in industry standards. If you make them thicker with agar, potentially the organism that you're trying to isolate or trying to cultivate will not grow as well because maybe the light might not penetrate it or things like that. So 1% is the industry standard to make for plates. And so what you do is you ask yourself, how many plates do I need for my experiment? And then once you figure that out, then you say, okay, about like 30 milliliters to 40 milliliters per plate is inside each plate. So you do a little multiplication. It's difficult. And you figure out the total volume that you need to use. So generally, 150 milliliters goes into those little tubes that we give you. And that generally makes about 7 to 8 plates, depending on how great you're pouring. So that's a calculation that you can do. And let's say you run out of LB agar. You can find it on our website. Oh, go figure. But we do give it in larger volumes. So when you do get a shipment of LB agar from us, it's in like 40 grams, I believe, which you can make a shit ton of plates from that. And it's generally cheaper. All right, so let's go over antibiotic selection. So there's an antibiotic selection kit. We sent you some kanamycin and some streptomycin. I cryptically put this as on the label, 30 milligrams per milliliter, and streptomycin as 33 milligrams per milliliter. And I gave you the molecular weight for these things. Okay, so let's do an example. So we want to make 100 micrograms per milliliter. So the reason why I say 100 micrograms per milliliter is because also this is also an industry standard, only because scientists are really lazy and we don't want to do too much maths. So 100 is a really good round number to work with, especially when you're working at like a microgram scale. You have to realize a microgram is, for example, 1 gram equals 10 to the 6th micrograms. So 10 to the 6th is a lot of zeros. So meaning you're making a very, very dilute antibiotic per plate. So we take 150 milliliters of the total solution, which is generally what we send you in those tubes, like the LB agar tubes. And then I would encourage you to try and make some antibiotic plates from these two separate entities. So let's do an example. Ampicillin is 100 milligrams per milliliter. So what you're going to do is you're going to just figure out what you're going to make for a stock solution. So the reason why we make a stock solution is because when you get ampicillin in powder form, it's really, really concentrated, as you can see. So, and you're trying to dilute this down all the way to like 10 to the 6th power. So we're going to do a series of dilutions. So first, we're going to take the powder form of ampicillin and then turn that into liquid form, which is your working stock. And your working stock should be 100 milligrams per milliliter. So generally, I don't like make a ton of working stock because all antibiotics can expire, especially so antibiotics also are temperature sensitive, meaning if you take ampicillin or any type of antibiotic in and out of the fridge, it can degrade and it can skew some of your results, which is not too great. So I would just take smaller volumes of it. Some laboratories that I know is they make like a really big stock and then they pretty much aliquot it into those little tiny 1.5 milliliter tubes that we sent you in bulk as well. So yeah, so we're going to do it in 10 milliliters of total solution. So this is kind of what I would set up how to like calculate it. And I would figure out, so for example, you see here 10 milligrams per milliliter equals X, which is what we're trying to solve for, out of 10 milliliters of total solution. That's how I'd set it up, which means that X would equal 1,000 milligrams. So you're like, okay, milligrams, great. You have to convert it to grams because your scale doesn't measure in milligrams, it measures in grams. So we convert it, and it is 1 gram. Cool. You have a scale, you have powder. Cool. Any questions so far? All right, we're good. So yes, 1 gram of ampicillin and 10 milliliters of water makes you 100 milligrams per milliliter of ampicillin stock concentration. So you can look online. Lots of antibiotics have different types of properties. For the purposes of this class, we're only using streptomycin and kanamycin, which can be dissolved in water. Some other random antibiotics will use like EDTA or maybe like a salt or an acid. So you just have to figure out online what's the best way to dissolve it. And you have to make sure you would dissolve it, or otherwise the molarity of the concentration will differ and it will not yield in what you want. So yeah, I would double check, look online if it's like an obscure antibiotic you're working with. But for the purposes of this class, it's all dissolved in water, which is pretty easy. All right, so going back here, we made only 100 milligrams per milliliter. So that's still like a couple more factors off from micrograms, right? So we need to dilute the stock concentration down to what you're actually going to be working with. So now you have to make another dilution. So now dilute it with this handy dandy, really cool equation. M1V1 equals M2V2. One side is going to equal the other side, which is awesome. So we're going to plug in for our V1, 150 milliliters, which is the amount of agar that you're totally using for 7 plates. Each tube that we usually give you is like, for like example, the LB agar is 150 milliliters total. So we're going to plug that in to our handy dandy M1V1, M2V2 formula. And you're going to do some maths, some cool maths. But first, you're going to have to convert milligrams to micrograms. And I handily gave you what the conversion is. Yep, that's it. So you're going to plug that back into the formula, as you can see right here. Do some maths, and then figure out for V2. So V2 ultimately was 0.15 milliliters. So you're like, crap, I can't measure in milliliters. I don't have a graduated cylinder, you know, small enough for this. Well, that's what a pipette's for. So we're going to convert milliliters into microliters. Yeah. Anybody have a number for me? Nada? Crickets?
Gave the answer.
Cool. All right, you get 150 microliters. So you add 150 microliters to your agar solution. So practically, I just want to give you like a practical sense of what we're doing is, so first you weigh out 1 gram of the antibiotic ampicillin. This is for ampicillin. And then you dilute it down to a stock concentration with water. And then now you have this like stock concentration, and now you have to add it to your agar media. So when you're generally making agar, as we practiced, you stick it in the microwave and you heat it up and you let all the agar dissolve. And at this point, you let the agar cool so you can like touch it, so you can pour it without potentially burning yourself. But right when you pour it, you always have to remember to add 150 microliters of ampicillin antibiotic inside of the agarose. Because if you add it while it's too hot, then the ampicillin will immediately degrade and will pretty much mess up your experiment. So, yep, once your agarose cools, then you add 150 milliliters or microliters, give a little swirl, and then you start to pour your plates. That way, all of your plates are uniform and they all have the concentration, the correct concentration that you have in your agar. Yeah. Oh my God. So any questions so far of like what I went through, how I did that in a practical sense? Oh my God. How do I end the screen share? Question? Comments? Tap water's cool. Yeah. Yeah, it's pretty straightforward, me and myself. Tap water, distilled water, it doesn't really matter too much, especially when you're working with antibiotics. Yeah, it doesn't really matter. Come and see us. What's your method for ensuring that everything is mixed properly? So I just give it a little swirl. As you know, when you shake up the agarose inside of the bottle, like really vigorously, then you'll get all those bubbles. You know, I wouldn't do that. I would add directly to the agarose and pipette 150 microliters of ampicillin, and then just kind of give a little swirl, and that's kind of put it on good faith that it's working. And you'll easily find out once you streak out your bacteria and nothing works. Yes, Seth, I will post my PowerPoint onto month 3. Come and see says, I know some labs use vibration machine, but we don't have that. You don't really need it. It's for lazy scientists. Alexandria Popova asks, what if we use cooking agar? Haven't really worked with cooking agar. From what I know, it's the same, like laboratory grade agar versus cooking agar. I don't know. Maybe you could do an experiment to see if there's any difference. Should be the same. I've actually ate agar in a lab once from a dare. We flavored it like cherry. It's pretty good.